the work I'm going to be presenting today is my postdoctoral studies, and the title of the talk is the life cycle of HER1 ion channels, and I'm going to be focusing on the mechanisms of gating and biogenesis. As an introduction, I'm just going to study for putting us in the physiological context, and I just want to mention that, can you see my pointer? No?
No.
Okay.
Now I can
Okay,
see.
I can use this one.
Okay.
I can use this one. Okay, so I just want to mention that the heartbeat rate and rhythm are controlled by action potentials that are traveling from the top to the bottom of the heart. These action potentials define the electrocardiogram waveform, as you can see by the matching colors. For example, the QT interval, which is highlighted in orange, is determined by the ventricular action potential, which is shown in the same color. Now, in turn, this ventricular action potential is shaped by an orchestrated arrangement of ionic currents conducted by different ion channels. The loss of any of these currents would affect the shape and the duration of the action potential. For example, the loss of the IKR current, which is critical during the repolarization phase, prolongs the ventricular action potential, lengthening the QT interval and leading to arrhythmias and even sudden death. These RKR currents are mainly conducted by heteromeric HER1A, 1B ion channels. These HER1A and 1B subunits are encoded by alternate transcripts of the same gene and differ only in their amino terminal region, with HER1A containing the PAS cap, PAS, and linker domains, while HER1B has a short and unique N-terminus. These subunits assemble as tetramers, of which HER1A homotetramers and HER1A 1B heterotetramers are functional at the plasma membrane. The HER1 channels are voltage-gated potassium channels that activate in response to membrane depolarization, transitioning from the closed to the open state to then rapidly inactivate. During membrane repolarization, then the inactivated channels recover from the inactivation and then slowly deactivate, transitioning now from the open to the closed state. This gating mechanism, of course, defines the shape of the HER1 elicited currents. And if now we look at these current traces and we compare the current elicited by the HER1A homotetramer and the HER1A 1B heterotetramer, we can see they are different. And this is, of course, due to differences in the biophysical properties of the conducting ion channels. One of these differences is the deactivation kinetics of the ion channels, or the closing. As you can see by this current decay that can be fitted to a second exponential function, the homomeric HER1A ion channels have a slower deactivation compared to the heteromeric HER1A 1B ion channels. But why is this different? If now we look at the model and the structure, the solved structure of the HER1A homomeric channel, and compare it to a putative model of HER1A 1B heteromeric channels, we can see that then the HER1B subunits that are lacking the PAS cap and PAS domain account for a difference. So it is almost intuitive to hypothesize that the lack of the PAS cap and the PAS domain, in the HER1B subunits might be responsible for these biophysical differences and therefore the deactivation kinetics of the heteromeric HER1A 1B ion channels. This was in some way previously established by the Robertson lab in a study in which they showed that the deletion of the entire amino terminal region of the HER1A subunit in the context of the HER1A homomeric channel accelerates the deactivation kinetics, as you can see here, compared to the wild-type channel in the current traces and in the quantification of the time constant of deactivation. This is also true if they only delete the first 16 amino acids corresponding to this disordered region here, or the PAS cap tip, as I'm going to be calling this region throughout the entire presentation. As you can see in the current traces, this deletion accelerated the deactivation, but interestingly, completely phenocopy the deletion of the entire amino terminal region, suggesting that not only the amino terminal region, but in a specific, the PAS cap tip is critical in determining the deactivation kinetics of the HER1A homotetramers. But what is the structural or molecular underlying mechanism to these functional differences? That's the question I decided to address when I joined the Robertson Lab. And based on my luck, like today I'm talking on the election day, I joined the Robertson Lab 2 weeks before the pandemic started, so it's my trending. When I was in the lockdown, I decided to study, like address this question. And during the lockdown, I just had the computer and the structure. And by analyzing these intracellular domains, I identified that in the interface between the globular path, the path cap helix, and the cyclic nucleotide binding homology domain, there is a three-dimensional hydrophobic nexus composed by apolar residues of the three mentioned regions. I want to highlight the point that most of the apolar residues are located at the globular path and the path cap helix. And these two domains have been previously involved in the deactivation kinetics. I hypothesized that the hydrophobicity of this intracellular nexus might tune the HER1A homotetramer deactivation rate. To test this hypothesis, and in collaboration with Whitney Stevens-Sostre, a PhD student I mentored during the first years of my postdoc, we decided to perturbate the nexus hydrophobicity in different ways to see what happens with the currents. Whitney initially characterized the current decay of the wild-type ion channel and the deletion of either the path cap tip, the path cap helix, or the entire path cap domain as a way to mutilate in some way the three-dimensional nexus. As a result, as expected, she saw that the wild-type ion channel has a slower deactivation that is accelerated either by the deletion of the path cap tip, the path cap helix, or the entire path cap domain, as you can see here in the quantification of the time constant of deactivation. But more interestingly, the setting substitution of these hydrophobic residues had an effect on the deactivation kinetics. And we substituted by setting residues because this is a hydrophilic amino acid that has an average size, so we are not introducing any aesthetic hindrance by purine these mutations. Now, when we analyze the currents elicited by the mutant ion channels, we observe that the setting substitution of the amino acids 15, 18, and 19 here in the middle of the helix accelerated the deactivation kinetics compared to the wild-type channels, but not the substitution of the phenylalanine 14 and 22. So to us, these results suggest that the path cap helix hydrophobicity determines the deactivation kinetics of the HER1A homomeric ion channels, and that the residues F14 and F22, which are at the limits of this helix, set the boundaries of the hydrophobic patch that is actively involved in the gating mechanism of this ion channel. But just to test if this is due to the hydrophilic properties of setting or just because, we introduce a random mutation in this context, we substituted the apolar wild-type amino acids, leucine and isoleucine, with cysteine or valine, which are amino acids more apolar than serine. And as a result of the same electrophysiological approach, we observed that when substituting these amino acids by valine or cysteine, there's no change in the activation kinetics compared to the wild-type channels. And of course, the deactivation is way slower when compared to the serine mutation. So we observed that only the hydrophilic amino acid was able to accelerate the deactivation kinetics of this ion channel. Therefore, it is indeed the hydrophobicity of this region what determines the HER1A deactivation rate. So far, I have just been talking about this hydrophobic patch in the path cap helix, but I mentioned at the beginning, and I identified a three-dimensional hydrophobic nexus. So to test the relevance of this hydrophobic nexus, once again, I went to the structure and then identified those apolar residues that are in close contact with the apolar amino acids in the path cap helix that affected the deactivation kinetics. And when I mean close contact, I mean 4.5 angstroms or less. As a result, as you might expect, I identified residues located in the globular path and the cyclic nucleotide binding homology domain. And therefore, of course, we mutated them by setting once again in order to affect the hydrophobicity of the region. As a result, we observed that the mutation of most of the residues accelerated the deactivation kinetics compared to the wild-type channels, suggesting that the hydrophobicity of the entire three-dimensional nexus is fundamental for the deactivation kinetics of the HER1A homometric channels. I also want to mention that the mutation of several of these residues has been implicated in different long QT related disease mechanisms. But once again, this is the question I always like to ask: what is the molecular mechanism underlying this functional thing we are seeing? To address this question, I perform molecular dynamic simulations, and then I just analyze the distance between the mutated residues in the PAS-CAP helix with respect the interacting partners either in the globular path or the cyclic nucleotide binding homology domain. As you can see here in this heat map, in the y-axis, I have the ion channels in which the analysis was done, which is the wild-type channel and, of course, the mutant. In the x-axis, we have the pair of residues under analysis, and by the color gradient, we have the distance between the residues, which gets lighter when the distance increases. As you can see by the animation and by the lighter color when having the mutation, we observe that mutating these residues increases the distance between the amino acid located in the PAS-CAP helix and the globular path and cyclic nucleotide binding homology domain. This analysis was also performed for the I19S mutant that accelerated the deactivation, observing once again the same. The distance increases with respect residues located in the globular path and the cyclic nucleotide binding homology domain. And finally, of course, we did it for the isoleucine 18, which only has interactive partners in the globular path, once again observing the same. So these results suggest that the mutations by setting are indeed disrupting the hydrophobic interaction network within this hydrophobic nexus that is responsible for slowing down the HER1A deactivation. But if the hydrophobicity of this region is actually what matters, then the closely related EAG ion channel that shows a fast deactivation then should lack this hydrophobicity. And to test this prediction, I went once again to the X structures and compared the composition of the globular path, PAS-CAP helix, and CMBHD domains between the ER1A channel and the EAG. And as a result, you can see that the EAG interface is way less crowded compared to the ER1A counterpart. And additionally, there is a reduction of 50% in the number of hydrophobic or apolar amino acids located in this region. So this result supports the hypothesis that the slow deactivation of HER1A ion channels is due to the high hydrophobicity of this interface. But a remaining question is how this hydrophobic nexus that is down here in the intracellular domains modulate the gating of the ion channel up here in the plasma membrane. I mentioned before that the deletion of only the disordered region is enough to accelerate the deactivation. So we hypothesize that by affecting this interaction network in the hydrophobic nexus, we are affecting the positioning of the PAS-CAP helix, which is covalently bound to this PAS-CAP disordered region. Therefore, by moving the helix, we are moving the PAS-CAP tip, and we are affecting the gating of the ion channel. To test this prediction, I analyzed the distance between the PAS-CAP tip and the S4, S5 linker belonging to the gating machinery. I used the first solved amino acid in the PAS-CAP tip and the aspartate 540 in the S4, S5 linker because this amino acid has been previously related to the deactivation of the ion channel. When comparing this distance between the wild-type channel and the mutants, we observed that for all the mutants, this distance increases, suggesting that our prediction was correct, that by affecting the interaction network in the hydrophobic nexus, we are displacing or decreasing the probability of the PAS-CAP tip to interact with the S4, F5 linker and therefore the gating machinery. So based on these results, we propose as a working model that the slow deactivation of HER1A ion channels is determined by the hydrophobicity of a three-dimensional nexus located at the interface between the globular path, the PAS-CAP helix, and the cyclic nucleotide binding homology domain. Now, perturbating this nexus hydrophobicity by introducing setting mutations promotes this rearrangement of the interaction network that results in a rotation of the intracellular gating ring with respect the pore of the ion channel. This rotation finally leads to the displacement of the PASCAP tip away from its binding pocket in the S4, S5 linker, which accelerates, based on the foot on the door hypothesis, accelerates the deactivation kinetics, which we finally see as faster current decays in our functional experiments. So when we talk about the life cycle of HER1 channel, as I decided to entitle this talk, we are talking here in the adulthood or the working years of the ion channel, and we are focusing on the gating, and what we are proposing that there is the hydrophobicity of an interface is critical in modulating the gating of these ion channels. But my interest in ion channels is not only on their structural function. I am also interested in their mechanism of biogenesis, like how they are formed and what is the physiological relevance of this process. And a key question to me is how the HER1A and 1B heterotetramers, like the two subunits, how they are finding each other and how they are forming these heterotetramers. So the question is, how are these heterotetramers formed? This question was initially addressed by the Robertson lab before I joined by a study in which they did the immunoprecipitation of the HER1A protein with an antibody targeting its amino terminal region. And as a result, they observed the couple down, of course, of the HER1A and 1B subunits, because this is an heterotetramer. But interestingly, the couple down of the encoding HER1A and 1B mRNAs, as you can see here in this RT-PCR gel. This result suggests that these HER1A and 1B ion channels might assemble co-translationally. But then how this putative co-translational complex is formed? Intuitive hypothesis is that the interaction between the nascent polypeptides might be the driving force that brings this entire complex together. To challenge this hypothesis, the Robertson lab introduced a mutation in the translation start site of the HER1B mRNA. So now we are in the context of having the two mRNAs, but only the HER1A protein, not the HER1B. They did the same experiment, immunoprecipitating the HER1A protein with the specific antibody, and as a result, once again, they observed that the two mRNAs came together, suggesting that the interaction between the nascent polypeptides is not required for the co-translational assembly of these ion channels. Thus, in summary, this study suggests that HER1A, 1B ion channels assemble co-translationally, but the interaction between the nascent polypeptides is not required for this process. If now we pay attention to these putative co-translational complexes and we think these interacting polypeptides are not required, then we have still two non-mutually exclusive mechanisms. We have the role of RNA binding proteins bringing the complex together, and we have the possibility of direct RNA-RNA interactions. In this talk, I'm going to be focusing on the second possibility, by hypothesizing that direct RNA-RNA interactions, which are determined by the RNA sequence and structure, promote the simultaneous translation of heteromeric HER1A, 1B ion channels. But before talking about direct RNA-RNA interactions, we just have to understand RNA, or at least the RNAs we are working with. Here I'm just showing the predicted secondary structure of HER1A and 1B mRNA, and the color gradient just indicate the base pairing probability. So you can see that in both structures, there are regions that are highly structured with high base pairing probability between the nucleotides. By comparing the two mRNAs, we see they differ in length as expected from mRNAs that are encoding subunits of different lengths, but they have a pretty similar normalized ensemble diversity. And what is normalized ensemble diversity, or net value, as I am going to be calling it throughout the entire presentation? You can imagine the RNA is a long thread, and that thread can be folded in multiple ways and remain stable. Of course, the longer the thread is, the more stable conformations that thread can have. And that's more or less the idea of the normalized ensemble diversity. It's a way to quantify the structural variability of an RNA, defined by the predicted number of possible stable structural conformations an RNA can occupy in the thermal equilibrium. Therefore, the higher this number, or close to 1, it means the RNA can have multiple conformations. Therefore, it's kind of a loose RNA, while the lower this number is, it means the mRNA is more stable in one conformation, therefore is more rigid. In this context, the HER1A and 1B mRNA have a similar net value, therefore they have the same probability of occupying a similar population of RNA structural conformations. Now, the interaction between the mRNAs and their structure is determined by the equilibrium between the intramolecular interactions that define the structure and the intermolecular interactions that define the association between the two mRNAs. These non-covalent interactions are mainly hydrogen bonds, base pairing, London-Barnett bus, cation pi interactions, the binding of some metal cations, etc. So by modulating this equilibrium, we can modulate, therefore, the association between the mRNAs and probably the co-translational mechanism of the HER1A, 1B ion channels if our hypothesis is true. To test whether HER1A and 1B mRNAs directly interact, we had to go in vitro because it's the only way that we can get rid of any biomolecule that can be bridging their association. To do this experiment, I did the independent in vitro transcription of the two mRNAs, and then I labeled them with spectrally separable fluorescent dyes and co-incubated them in a buffer containing magnesium chloride, spermine, and potassium chloride, all of them known to stabilize the RNA structure, and I did the incubation for 3 hours at 37 Celsius degrees. After that time, I just did the imaging at a really low resolution. So this the resolution by the Abbe equation here is about 600 nanometers. So we understand this PONTA as complexes, not like single molecule RNAs. I decided to play with the potassium concentration just to see what is the effect of the major monovalent intracellular cation in the association between the mRNAs. The association between the mRNAs was determined via their colocalization and quantified by the Pearson correlation coefficient of the signal overlap. As you can see here, for almost all the conditions tested, the association between the Pearson correlation coefficient is higher than 0.5, suggesting the mRNAs directly interact. But this interaction might be just due to random stickiness or random interactions between these naked RNAs. So to test for specificity, we substituted the HER1A mRNA by phospholamban. And why phospholamban? Phospholamban is an mRNA that encodes the phospholamban protein, which is an ER membrane protein that is expressed in cardiomyocytes and is extremely relevant in the excitation coupling of these cells. Besides, the phospholamban mRNA has a similar length to the HER1A and 1B mRNA and a similar net value, suggesting it has the same probability of opening and closing and interacting with other mRNAs. Therefore, we thought it would be a good negative control. After performing the heterotypic in vitro reconstitution of PLN with 1B, we observed that for all the tested conditions, the colocalization between these two mRNAs is markedly decreased compared to the HER1A, 1B association, suggesting that HER1B has a higher ability for HER1A compared to phospholamban. So these results suggest that the HER1A and 1B mRNAs directly and specifically interact in vitro. But now, if we look at the effect of potassium on this association, we can see kind of a biphasic effect in which at lower potassium concentrations, the association increases, and then when the potassium concentration keeps going up, the association is affected. So these results suggest that the heterotypic associations between the mRNAs is modulated by the potassium concentration. But why is this happening? by reading, we came out with the concept that monovalent metal cations, despite having the same charge, have different effects on the structure of RNA. For example, when talking about the G quadruplex, which are RNA structural motifs that are formed by quartets of guanines, we observed that potassium and sodium have different stabilizing effects. And this was shown in this study in which they have two mRNAs that are known to form G quadruplexes, and they are measuring the melting temperature of those mRNAs as a proxy for mRNA stability. And as you can see here in red, when increasing the potassium concentration, the stability of the mRNAs increases, but nothing happens when changing sodium. So this suggests that the metal cations have different stabilizing effects on the RNA, and this is due to different properties such as the charge density. So now, to know if the biphasic effect we saw with potassium is due to just the positive charges or is due to a specific effect on the RNA structure, we substituted potassium by sodium, observing as a result that the biphasic effect was completely lost. So these results suggest that the potassium effect on HER1A 1B heterotypic interactions might be related to the stabilization of certain structural motifs that increases the intramolecular interactions, therefore affect their association of these mRNAs with any other molecule. To test this rationale in an orthogonal way, we then decided to do mutations to the RNA. And then in collaboration with Ian Same from the Amy Gladfelter's lab, we designed 3 synonymously mutated mRNAs that, of course, encode the HER1A protein. These mRNAs differ in their structural diversity, having a higher net value, a similar net value, and a low net value. As you can see by the base pairing probability, that mRNA with the lowest net value has a huge base pairing probability, so it must be a really rigid mRNA. Just to double check this synonymous mutation encoded the HER1A protein with the Western blot and observed that the protein was recognized with an antibody targeting the HER1A specific amino terminal region and the common pan-HER region between HER1A and 1B mRNAs. Now, to test the hypothesis that affecting the RNA structure might affect the association between HER1A and 1B mRNAs, we first went to bioinformatic predictions of the thermodynamic of this interaction. As a result, we did a relationship between the free energy of binding and the net value or the rigidity of the RNA, observing a linear relationship that suggests that the lower the net value, the higher the delta G, therefore the lower the probability of interactions between these two mRNAs, which is in agreement with our hypothesis and our observation with potassium. To test this prediction, we now did the in vitro reconstitution of the mutant mRNAs with 1B and compared their colocalization quantified by the Pearson correlation coefficient. And as a result, you can see that only for the mutant with the low net value, the association between HER1A and 1B mRNAs was affected, which is in agreement with our thermodynamic predictions. Therefore, these results suggest in general that decreasing the HER1A structural diversity decreases its association with 1B. So far, via in vitro reconstitution experiments, we have shown that HER1A and 1B mRNAs specifically interact and that their interaction is modulated by potassium concentration that affects their intermolecular association. Also, we have shown that the RNA structural diversity affects the heterotypic interactions between HER1A and 1B mRNAs, and we rationalize that the higher base pairing probability observed in the low net mRNA is what increases the thermodynamic barrier to undergo intermolecular interactions with 1B and therefore affects the association. But this happens in vitro. What happens in cells? To address this question, then I just did the independent in vitro transcription and labeling of the mRNAs and then co-transfected them in HeLa cells once labeled and just analyzed the colocalization between the signals by measuring the distance between the center of the puncta that we can observe. As a result, we observe once again that HER1B colocalizes markedly more with HER1A than with PLN, suggesting the higher ability of this mRNA for HER1A. And to test whether these interactions were dynamic or not, we use as a rationale that the dynamic of the interaction might be related to changes in the colocalization that might reflect the equilibrium between the unbound and bound mRNAs. As you can observe in this heat map, in which in the y-axis I'm showing the cells, in the x-axis I'm showing the time, and then by the color gradient I'm showing the colocalization of the mRNAs being greater when they're redder. We can observe just by looking at it that the colocalization between the HER1A and 1B mRNAs was significantly higher compared to phospholamban in all the cells tested for all the times. But additionally, if we look at the change in the colors, which would mean the colocalization change, we observe it is more dynamic in HER1A, 1B than in phospholamban, as you can see in the quantification, but also as expected, because two mRNAs that initially don't interact don't have any dynamic interaction. Thus, these results suggest that the heterotypic HER1A and 1B mRNA interactions are specific and dynamic in HeLa cells. But what is the role then of the RNA structure on the association of these two mRNAs in the cells is the same. We have now RNA binding proteins. We have now all the cellular context. So once again, I did the independent in vitro transcription and labeling of the RNAs and co-transfected them in HeLa cells and analyzed their colocalization. And as you can see here, all the mutant mRNAs showed a decreased association compared to the wild-type channel, different to what we observed in vitro, in which only the low net value mRNA showed a decrease, suggesting that in this case, not only the structure, but also the RNA sequence might have an effect on the association between the RNAs. But anyways, the mRNA that showed the biggest decrease was the mRNA with a low net value, suggesting that, again, in cells, the mRNA sequence and structure determines the association between HER1A and 1B. So now that we are in cells, and we know that the structure affects the association between the two mRNAs, thus affecting these mRNAs interaction affect the co-translational association of the encoded ion channels, that's the natural question to ask. So to address this question, we came with the approach of adding spectrally separable fluorescent proteins to the HER1A and 1B mRNAs, and then we are gonna use their encoding mRNAs, fluorescent protein tags to the HER1A and 1B proteins, and then we are gonna transfect the HER1A and 1B mRNAs. So once again, I did independent in vitro transcription of the RNAs, the labeling, and co-transfected them in HeLa cells, and 48 hours later, we are going to analyze what is happening in the ER here. So when analyzing the translational complexes of HER1A, we have different possible mechanisms. The first one is just the individually translated HER1A subunit, which would be characterized by the colocalization between the HER1A mRNA and protein. But we can also have the diverse co-translational mechanisms. It could be a simultaneous mechanism that is characterized by the two proteins undergoing translation. Therefore, we can see it as the colocalization between the two mRNAs and the two proteins, or we can have a sequential mechanism that is characterized by the association of a fully translated protein with another one that is undergoing translation. And in this case, this one would be identified by the colocalization between the HER1A mRNA and protein plus the HER1B protein. When analyzing all these complexes in the cells, we observe that for all the mutants, most of the HER1A mRNAs are undergoing co-translational assembly with HER1B, while a minority is just undergoing individual translation. But anyways, when comparing between the mutants, we observe that only the mutant with the low net value decreases the percentage of mRNAs undergoing co-translational association, therefore increasing the percentage of individually translated HER1A proteins. But now, if we go inside all these co-translational complexes and divide them between simultaneous or sequential, as I mentioned before, we see that for almost all the mutants, the simultaneous co-translational association of the proteins is the majority mechanism with a lower amount of the sequential process. But when substituting the HER1A mRNA by the low net value, which affects the direct RNA-RNA interactions, we see the simultaneous mechanism is disfavored. Instead, there is a promotion of the sequential mechanism, suggesting that by affecting the direct RNA-RNA interactions, we are affecting the mechanism of co-translational association of the mRNAs, and then the cell finds a redundant pathway by which the cell guarantees there's going to be the formation of these heterotetramers. And this is consistent with the fact that when we analyze the colocalization between the HER1A and 1B proteins in the cells with the different mutant mRNAs, the colocalization is the same, suggesting that by balancing the equilibrium between a simultaneous and sequential association, the cell guarantees the formation of the heterotetramers, which are known to be essential for the cardiac activity. So as a working model of this second part, we propose that the HER1A and 1B ion channels assemble co-translationally, and that in native conditions, including cardiomyocytes that I didn't show here, but we did it, direct RNA-RNA interactions contribute to the simultaneous co-translational association of the HER1A and 1B subunits as the majoritary mechanism. But by affecting the RNA structure, like increasing its structural diversity and therefore affecting their association, we then we reduce the number of simultaneous co-translational complexes, promoting a sequential pathway that guarantees also the formation of the heteromeric ion channels. Thus, as putting it in the context of the life cycle of HER1 channel, in this second part, we talk about the biogenesis and propose that direct RNA-RNA interactions promote the simultaneous co-translational association of HER1A and 1B. So in this way, we are able to talk about the gating and biogenesis of these ion channels and either the homotetramer or the heterotetramer. As a closing remark, I want to thank my collaborators in both projects. First, Whitney, Danielle, and Lucy, which were the major collaborators in the structure function paper. And then to Annabelle and Lucas, these two wonderful undergrads, which are my major collaborators in the biogenesis mechanism, and ER from the Glyphater lab. Of course, to Gail for her wonderful mentorship, and all the current and past members of the Robertson lab. And my funding sources during the postdoc, including Gail Sarowan initially, then T32 from the Cardiovascular Research Center, and then currently my K99 R00 supported by NHLBI, and the administrative support that helping me to get on time after having my beautiful, wonderful baby, and just to Black on biophysics for all the support and networking. As Tamer mentioned before, I'm in the job market, so if you happen to like anything I said here and you happen to see my CV, just please read it to see if you like it, and I'm happy to take any questions.
Hi, Liz. Thank you. Fantastic. So for those that don't know, if you would like to add any questions, you're welcome to add them to the Q&A box now. Or if you would like to ask your question in person, you could raise your hand and we can unmute your microphone. So thank you, Liz. That was a fantastic presentation. We've got a couple of questions already. The first one is from David Colquhoun, who says, nice work, but it raises a problem which is very common in this sort of work. The macroscopic time constants, what you observe, are all dependent on every one of the underlying transition rate constants, elements of the Q matrix. This makes observed time constants hard to interpret in mechanistic detail in the absence of a specified mechanism.
I mean, yes, we don't know in detail what is the allosteric pathway that is guiding this. We cannot discern which is the amino acid or how the hydrophobicity is guiding this process. We can just say that mainly in a quantitative way, that by affecting this pathway, we affect the activation of the ion channels. We try to go to some structural details with the molecular dynamic simulations, but for sure, we cannot say an actual mechanism of the activation of these ion channels.
I think that helps you. Thank you. Do those mutations affect the activation rate?
We haven't tested that.
Okay, next question from Julius Ashot Kozak. who notes that you have 10 millimolar spermine present, which has a higher charge density than potassium or sodium. Lowering spermine might tell you if the potassium effect is really due to charge density or not.
We did the experiment with spermine. Let me see if I find it here, because in a previous, can you see here?
Yep.
So in the previous paper, where the Robertson lab showed the Translation Association of the encoding mRNAs, if you see here at the IP part, when you have the co-expression between the two subunits, you have this coupled down suggesting the translational association. But when you combine the HER1A, when you combine lyses of cells expressing HER1A and 1B independently, this association doesn't happen anymore. So this suggests this might be in the cellular context. And I have been telling you all this time that they interact in vitro. So what is going on here? So spermine has been used in this world of RNA condensates as a crowding agent. And what we try to hypothesize is that the crowded environment of the cell is required for this association. So what we did was just to play with the spermine concentration going from 0 till 10, which is kind of a range in which a spermine can be found, even higher sometimes. But when increasing a spermine concentration just to 5 millimolar, we see a saturating effect. So we don't think. And also in the potassium experiments, spermine is kept constant. So whatever change we are seeing in the potassium experiment is due to the change in potassium and not actually spermine, because we are keeping it the same. That's what we reasoned.
Do you think these changes in cations might be dynamic? It might regulate the co-translation of these proteins in vivo? Big question.
Yeah.
Yeah, have you ever tried incubating cells in Warbane, or however you pronounce it, to block the pump and see if you decrease?
No, I haven't tried that. And the challenge here is kind of how to set up like a calibration curve of changes in potassium concentration where these ion channels are undergoing translation, because it doesn't have to be change in the potassium concentration in the entire cell. It's just enough to be in the region when the co-translation is happening for having an effect on the association of the mRNA. So yeah, I'm still struggling with a way to do this, but hopefully I will find it.
Yeah, brilliant. Okay, moving on. Damien Bell, lovely work and story, Dr. Flores Aldama. Congratulations to you in the lab. The co-translational association biogenesis is fascinating. Do you think this is a common process for ion channels or other membrane proteins?
Yes, so this is what hopefully my future lab is going to do. So I'm going to be focusing on the telomerization mechanism of ion channels, trying to use the characteristics of the structure of the ion channels as a way to predict which is the co-translational mechanism, or if there is a co-translational mechanism mediating this. But based on this work and other studies that have been done in membrane-associated proteins, I think this is pretty common indeed. The 70% of the human proteome is predicted to undergo co-translational association.
Great. A second question from Julius Kozak. Maybe you covered this. Does hERG have a signal peptide for the ER?
What? I didn't get that.
Does the hERG channel have a signal peptide for the ER?
Yes. Yes, indeed, when HER1B is expressed by itself, it has a retention signal that doesn't allow the ion channel to traffic to the plasma membrane. That's why the HER1B homomers are not functional. They just get retained. And when you co-express HER1A with this HER1B that are already retained in the plasma membrane, then is when the ion channels can traffic to, that have been retained in the ER is when the ion channels can traffic to the plasma membrane. I'm sorry, I'm being really random talking here.
Got it. Okay, next question from Robert Isle. I'm interested in your observations that mRNA processing was impacted directly by potassium concentration independent of membrane potential. I study ion transport and abundance in T lymphocytes and see it's difficult to explain difficult to explain phenotypes that are due to intracellular potassium independent of membrane potential. How do you propose that potassium concentration itself is impacting mRNA processing or the interaction of the two subunits?
To me, if we want to relate it to the membrane, it's pretty interesting to me that these are also, I don't know, I expect that this effect of potassium on the RNA structure is relevant for all the mRNAs. But in this case that we know it is, these are potassium selective ion channels. could this be a, retroalimentation, like a feedback process by which there are changes in the structure of the RNA, and then it says, okay, there's no more translation because we have enough ion channels at the plasma membrane? It might sound crazy, but we don't know in case the translation is happening close to the plasma membrane because we know ER is everywhere. It can actually affect the expression of these heteromeric ion channels.
Sounds like T lymphocytes might be a good cell to look at then.
Yes.
If we have any more questions, feel free to answer, stick them in the chat. I've got one thing that occurred to me. So in the sequential translation, what's stopping the second ribosome from starting translation? So it's just it's bound to the mRNA and it's just waiting to initiate translation?
So we have, see, this sequential translation to me is really unfavorable for this kind of proteins that are highly intertwined, because it means that one subunit has to be held by a lot of chaperones to stay in this structure to then wait for the second one to be built. So it makes more sense, for example, ion channels like ATP, as I mentioned, like I was talking to you before, that the interaction is just on the C-terminal of one of the subunits. So one of the subunit can be completely formed, completely folded, and then interact with the other one that is coming through. I don't think that ion channels that the pore is formed, by intertwined subunits are gonna go, this sequential mechanism.
Right. Thank you. Okay. Well, I think we are done with questions. If any, go on, Layla.
Okay, since we have a little time, I'd just like to ask a bit of a nerdy question here. So there's really fascinating work. I was wondering, in the first section of your talk, where you talked about the back tip domain and its effect on function, I was wondering, is it possible to purify that region of the protein and use it like as a ligand, basically, as a modulatory peptide?
So that has been done before. So it has been done, like, not even just that region, just introducing the pass domain entirely, just is enough to bind there and slow down the deactivation. Like, it's wonderful. And it also called my attention from the molecular dynamic simulation point of view, the fact that in the structure, this domain, like the pass domain, is not covalently attached to the rest of the ion channel. And even when it is not covalently attached, it remained there throughout the entire simulation. So these interactions are really a thing, like they are really strong.
Okay, cool.
Thank you.
Lisandra, I understand the logic for looking at cations, but have you tested whether chloride changes any of the mRNA interactions? Because that intracellular chloride concentrations can range quite a lot from cell to cell. So I wonder whether that could be a...
Yeah. I mean, the idea of cations stabilizing the RNA structure is just to decrease the repulsive between the major, negatively charged of the backbone. So I guess if we increase now chloride, we are just kind of open more the RNAs compared to, so when we have potassium, it tends to close the RNA structure. Maybe when we have chloride, that's cool. We can, open it a little more. Maybe they would interact a little more. Yeah, I don't know. That's cool. Thank you.
Okay. Fantastic. Thank you for your time. That was a fantastic presentation, and thanks for all the questions and participation and the attendance, everyone in the audience. The next seminar is, I'm looking at the schedule, is on the 3rd of December, and will be Rianne Manville from UC Irvine. So hopefully see everyone there. Thank you again, Lisandra. Best of luck with everything, and we'll see you all soon.